Hi everyone, I'm Nabaraj and I'm presenting my project on the extraction, quantification and evaluation of antioxidant and anti-inflammatory effect of Clove's derived compound named Eugenol on H2O2-induced SIY neuronal cells. So basically, Cloves contain a compound Eugenol which is known as its antioxidant and anti-inflammatory properties. Oxidative stress, like the kind caused by hydrogen peroxide, can damage cells, especially neuronal cells. There is also a gene called COX-2 that is usually involved in anti-inflammation during this kind of stress. So what I wanna see was, can Eugenol actually helps either handle or recover from this stress. So the goal was to extract the compound Eugenol from the clove buds, test its antioxidant properties, and then see how it affects cell viability and gene expression in stress cells. To do this, I extracted the cloves compound using Sox-led extraction method and then quantify the compound through the GC. Then I checked its antioxidant properties using two different assays, FRAB and FNC, where for the cell experiment I used SIY neuronal cells and then tested its approaches. Pre-treatment where cell get Eugenol before, and then post-treatment was where Eugenol is added after the stress. Then I measured the cell viability using MTTXA and furthermore look at its gene expression using real-time qPCR. Talking about the results, the extract shows a strong antioxidant activities which was expected, and then confirmed that cloves is rich in phenolic compounds. In cell viability, when I looked at it, the hydrogen peroxide reduced the cell survival rate, so the oxidative stress was clearly working. But the interesting part was post-treatment actually works better than the pre-treatment. So instead of protecting the cells beforehand, Eugenol seems to help them recover after damage. That's a bit unexpected, but it's usually pretty interesting, although this wasn't perfect consistent across all the concentration. So the effect likely depends upon the doses and then different conditions like time. For the gene expression, I looked at COX-2. There was amplification but also some non-specific bands, and overall there were not any significant difference between the groups. So overall, oxidative stress was successfully induced, and is also some positive effect, especially after post-treatment. Even the induced time was just 2 hours. This suggests it might be more useful for recovery than the prevention, but based on the gene expression results, the anti-inflammatory effect couldn't be clearly confirmed. For the future works, I'd focus on optimizing the stress condition and then improving PCR accuracy Also, since the post-treatment shows better result, it would be interesting to see how Eugenol helps in recovery pathway. Here are some references, and at the end I'd like to thank all my professor for their help and then support during this research project. Thank you.